Hello students, today we will discuss diversity in protein phosphatases. Protein phosphatases are several classes of enzymes that catalyze the hydrolysis of phospho-amino acids within peptide or protein, thus resulting in dephosphorylation. Originally considered as boring enzymes, it is clear by now that phosphatases are playing dynamic regulatory and decisive roles. They are obligate counterparts of protein kinases. As we have already discussed in our previous lecture, protein phosphorylation is a common regulatory mechanism in all other eukaryotes, bacteria, and archaea, in which protein kinases, if you remember, transfer the gamma phosphate group of donor ATP to the acceptor side chains, while protein phosphatases dephosphorylate the phosphoproteins, right? The intracellular phosphorylation level of protein is catalyzed by protein kinases. Just look at this diagram. Substrate, protein kinases, and protein phosphatases here. Here, protein kinases they catalyze the phosphorylation level of proteins. Phosphorylation means addition of phosphate group to substrate. And this phosphate group has been taken from ATP, gamma phosphate group of ATP, right? On the other hand, protein phosphatases, they remove this phosphate group from the substrate, means dephosphorylation. In this way, coordinated control of kinases and phosphatases activities provide the cell with capacity to rapidly switch proteins from the phosphorylated to dephosphorylated state to meet the different physiological demands. Protein phosphatases are involved in many fundamental physiological events like growth, proliferation, survival, differentiation of cells, stress response, hormone action, ion channel activities, then neurotransmission, protein synthesis, gene expression, and various metabolic pathways, right? Now, important point. Remember, the human protein kinome and phosphatome comprise 518 protein kinases and 189 protein phosphatases, respectively, while about 1125 protein kinases and 150 protein phosphatases have been reported in Arabidopsis, a model plant. Another important point, as most protein kinases possess beta sheet N-terminal to bind ATP and alpha helical C-terminal to bind substrate. In contrast, protein phosphatases exhibit diverse superfamilies with different 3D structures, active sites, and hydrolytic mechanisms, right? Depending on their substrate specificity, mechanism of catalysis, and inhibitor sensitivity, eukaryotic protein phosphatases are classified into 4 major classes. Number 1, the phosphoprotein phosphatases, that is PPP, metallo-dependent protein phosphatases, PPM, protein tyrosine phosphatases, PTP, and last, aspartate-dependent phosphatases. First two, PPP and PPM families are serine threonine specific phosphatases, while third one, PTP family, is tyrosine specific. PPP and PPM families share a similar structural fold, indicating a common catalytic mechanism. First, we'll start with phosphoprotein phosphatases, that is PPP. This family of protein phosphatases consists of PP1, PP2A, PP2B, PP4, PP5, PP6, and PP7, right? All the PPP enzymes have same basic architecture with a series of conserved domains and motifs. Just look at this diagram. This is all the members of phosphoprotein phosphatases family. PP2A, PP4, 6, 1, PP2B, PP5, and PP7, right? All members of this PPP family has 3 highly conserved signature sequence motifs. These 3, GDXHG, GDXVDRG, and GNHE. Now, what is this GDXHG? X, HG. Here G is glycine, D is aspartic acid, X is amino acid, H histidine, V valine, R arginine, N asparagine, and E glutamic acid. All these amino acids coordinate two divalent metalloids, either magnesium or manganese, or iron and zinc ions in catalytic center. First is PP1. PP1 exhibit highly conserved sequence and structure among eukaryotes, with the plant and animal PP1s showing more than 70% identical residues in the core catalytic domain. Just look at this diagram. This is catalytic core domain, right? Each functional PP1 enzyme consists of catalytic subunit and a regulatory subunit. Catalytic subunits adopt a compact alpha-beta fold with beta sandwich wedged between two alpha helical domains. Now, one thing more. PP1 is a single domain hub protein, right? PP1 catalytic subunit binds to regulatory subunit, resulting in functional enzyme. PP2A protein phosphatases 2A. It is an important serine threonine phosphatase that plays a critical role in cellular physiology, including cell cycle, cell proliferation, development, and regulation of multiple signal transduction pathways. PP2A coenzyme comprises 65 kD scaffold subunit and 36 kD catalytic subunit to gain full activity towards specific substrate. The PP2A coenzyme interacts with variable regulatory subunits to form a heterotrimeric holoenzyme. Then PP2B, also known as calcineurin, consists of a catalytic subunit, calcineurin A, that is CNA, and a regulatory subunit, also known as calcineurin B or CNB. CNA contains N-terminal phosphatase domain, followed by CNB binding helical domain. Then a calcium chameleon binding motif and an autoinhibitory element, right? Calcineurin is inactive alone and only gains phosphatase activity upon association with calcium chameleon, right? And it plays important role in numerous calcium-dependent biological processes, including neurodevelopment and memory, immune response, cardiac hypertrophy, signal transduction, and muscle development, etc. Right? This is the structure of PP2B. All these motifs are same as we have discussed previously. In addition, these two domains BBH and CBD and EN are here in the structure of PP2B. Then PP5, it regulates cellular proliferation, differentiation, migration, survival and death, and also DNA damage repair. Important role that this PP5 plays in hormone and stress-induced signaling, right? PP5 contains a regulatory domain at N-terminus. This is N-terminus part and this is C-terminus part. Here, a regulatory domain that is TPR domain. TPR, tetra-tricopeptide repeat domain. It is present at N-terminus, right? And phosphatase activity of this free PP5 is suppressed because this TPR domain and a C-terminal helix alpha J together maintain PP5 in an auto-inhibited conformation. Next, PP4, PP6 and PP7. Catalytic subunits of PP4 and PP6 are closely related to catalytic subunit of PP2A, right? Catalytic subunit of PP6 is thought to form a heterotrimeric holoenzyme with a site 4 associated protein SAP domain containing scaffold subunit, right? And an ankyrin repeat subunit that likely serve as a regulatory subunit, right? Catalytic subunit of PP4 also associates with its own regulatory subunit R1 and R2 to form distinct complexes. Now, In contrast to pp4 and pp6, pp7 appears to be a unique to plants, right? pp7 contains 3 insertion in its phosphate domain, and the recombinant pp7 protein gained phosphatase activity only after cleavage of the longest insertion, suggesting an autoinhibitory role, right? Now, pp7 was shown to interact with calcium and camodule, but in contrast to calcineurin, this interaction appears to inhibit the phosphatase activity of pp7. Now, next type of protein phosphatases is magnesium-manganese ion-dependent protein phosphatases, ppm. This family includes protein phosphatases dependent on manganese-magnesium ion, such as pp2c. pp2c is an example of serine threonine specific phosphatases, right? Means magnesium and manganese dependent protein phosphatases. Now, both PPP and PPM. Metal ions here play a catalytic and central role through the activation of water molecule for dephosphorylation reaction. Here, active site motifs, you can see here, this is the structure of pp2c. Here, core structure of ppm is the 300 residue ppm type phosphatase domain that catalyze the dephosphorylation of phosphoserine and phosphothreonine containing proteins, right? Eukaryotic pp2c have variable catalytic domain at either N-terminus or at C-terminus, right? Now, this catalytic core domain contains 11 characteristic subdomains. Third is phosphotyrosine phosphatases. This is the largest family of phosphatases gene and are defined by their active site signature motifs CX5R, right? The complement of human PTP genes can be divided into 3 distinct families based on the sequence of their PTP domains. First is class 1 PTPs having 100 genes, class 2 PTPs having single gene only, and class 3 PTPs with 3 genes. Class 1 PTPs constitute the vast majority of human PTP genes and can be divided into classical phosphotyrosine specific PTP and VH1-like or dual specificity protein phosphatases, right? DSP or DUSP subfamilies. DUSP subfamilies with 63 genes is the most diverse group in terms of substrate specificity. So this group includes MAP, MKPs, DSPs, PRL, PTENs, inositol 1,3 triphosphate, and inositol 1,3 biphosphate, right? And now the last type, aspartate-based protein phosphatases. Member of FCP and SCP family rely on aspartic acids of the sequence motifs DX-DXTB for phosphatase activity, right? So here, signature motifs of this family is, you can see here, DX-DXT, which is located at C-terminal to the first beta strand of the conserved core domain, right? Now, the only known substrate for FCP and SCP is the C-terminal domain of RNA polymerase II, which contains tandem repeats of serine-rich heptapeptide, right? So, just look at this diagram. This is the structure of SCP1 and this is FCP, right? So, here FCPs, but not in SCPs, contain BRCT, means BRCA1 C-terminal domain, like domain that is C-terminal to the FCPH domain, right? So this is all about protein phosphatases. If you have any question, please write in the comment section so that I can answer, right? So if you like this video, make sure to subscribe and for more. Thank you so much.